organized by the Department of Pharmacy, Faculty of Mathematics and Natural Sciences, Universitas 11 Maret Indonesia, as part of program Kreativitas Kompetisi Kampus Merdeka or PKKM. I'm Erna Wulandari. I'm moderator of today's webinar. I would like to extend my greetings to the Dean of our faculty, Dr. Andes Harjana, Master of Science PhD, the Vice Dean of Academic Research and Student Affairs, Professor Fenty Suryanti, Bachelor of Science, Master of Philosophy PhD, the Head of Pharmacy Department, Dr. Rerum Naturalium Pharmacy Sabtono Hadi, Bachelor of Science and Master of Science, as well as the Head of the Program Studi Profesi Apoteker, our representative of the industrial partners, and our distinguished speakers, Professor Alfi Khatib and Assistant Professor Siti Hadijah Samsudin. Today's webinar marks the launching of PKKM webinar series, and we will have two exciting topics for today's webinar. First is the metabolomics in herbal medicines development, and the second one is the development and commercialization of herbal medicines. Just a little housekeeping before we get started for today's webinar. I will first read out the agenda for today. Firstly, we have the opening, followed by the formal opening speech from the representative of the Dean of the Faculty of Mathematics and Natural Sciences, Universitas 11 Maret, followed by the presentation of today's topic. Each talk will be presented for 1 hour consecutively before we come to the panel discussion for 30 minutes at the end of the webinar. The last event is closing, that includes the photo session, and I invite everyone to join our photo session. Before we start with the first event, I'd like to remind everyone to turn off your microphone to make sure that the talk can run smoothly. And I would like to remind us as well that in the case of accidental voice on your microphone, the host will kindly turn off your microphone and we have your cooperation. And if you have any question at any time during the talk, please write them in the chat box. I will ensure that all the questions will be brought up during the panel discussion at the end. Otherwise, please ask your question directly to the speakers. Another important point is that please ensure you fill the attendance form that will be shared in the chat box. Now, without further ado, we will turn the time over to the Vice Dean of Academic Research and Student Affairs of the Faculty of Mathematics and Natural Sciences, Universitas Negeri 11 Maret, Professor Fenty Suryanti. Please, on behalf of the Dean of the Faculty, the time is yours. Prof. Fenty, your microphone is still on mute. Yeah.
Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi semuanya. Tadi pagi hari ini sibuk sekali karena akan ada acara materi kaitan program tahun mana pada pagi hari kita akan ada speaker Associate Professor Dr. Sebelumnya saya mengucapkan terima kasih kepada Kaprodi S1 Farmasi dan juga Kaprodi SPA atas undangannya, atas keberhasilannya untuk menangkan program kampus. Kemudian saya ucapkan terima kasih juga kepada Bapak Ibu dosen farmasi maupun SPA dan Bapak Ibu dari mitra yang juga sudah hadir di saya ucapkan terima kasih juga kepada semua dosen fakultas di SPA dan siswa yang di nasional juga dengan nasional ini diikuti dengan kegiatan internasional yang lain fakultas SPA Associate Profesor yang sudah bersedia untuk menjadi kita kedepannya kolaborasi dan sangat juga kegiatan kajian saya yang tanam lagi saya atas nama Pak Dekan mengucapkan terima kasih semoga kegiatan ini lancar dan siswa dosen bisa mengambil manfaat yang besar. Dan semoga yang bisa saya sampaikan di mohon maaf ini saya di luar ruangan jadi agak
Waalaikumsalam. Thank you, Prof. Henry. It is also our hope to continue this webinar series with more sustainable and continuous partnership with our international partners. Thank you for the support from the faculty. Next, we will enter the main event for today's webinar. Just a quick reminder again for all the attendees, if you have any questions during the talk, please type them in the chat box. And also please fill the attendance form later when we share the link for the attendance form. First is the lecture from Professor Afi Khatib in the Metabolomics in Herbal Medicines Development. Professor Afi Khatib obtained his Bachelor of Science degree from Bogor and Agricultural University in Indonesia. And then he was awarded the Thales Scholarship from the Dutch government for his Master and PhD in Pharmacognosy in Leiden University in the Netherlands. He also won the Dutch New Venture Competition Round 2 based on his PhD work on iso-alpha acid purification in 2006. He then continued to postdoctoral position before moving to University Putra Malaysia in 2008 as a senior lecturer. He then joined the Department of Pharmaceutical Chemistry, Kuliah of Pharmacy, International Islamic University Malaysia, or IIUM, in March 2013. From then on, IIUM has awarded him numerous awards, for example, the Excellent Research Achievement Award for consecutive years from 2017 to 2022. His research on the standardization of natural products using metabolomic approach and purification of bioactive compounds from Malaysian plants has led him to securing multiple research grants, especially from Malaysian government. Until now, he has published more than 130 articles, all in peer-reviewed international journals, and that has manifested in the very high H index of 30 and international patents. It is truly an honor to have you here, Professor Afi Khatib. Now, please, the floor is yours.
Thank you very much, Dr. Dr. Erna Wulandari, for introducing me to the participant. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi taala wabarakatuh. Alhamdulillah, rabbil alamin, wassalatu wassalamu ala asyrafil anbiya'i wal mursalin wa ala alihi washabihi ajmain. Rabbi shrah li sadri wa yassir li amri wa hlul 'uqdatam min lisani yafqahu qawli. Respected Professor Farti Suryanti, as the Deputy Dean of the Academic and Research Faculty of Mathematics and Natural Science, WNS, and also respected Dean Faculty of Mathematics and Natural Science, WNS, respected Chairman of the webinar, respected top management of the faculty, also the respective prof, doctor, brother, sister, and of course, respected moderator, Dr. Erna Wulandari. So I would like to acknowledge the organizer to give me an opportunity to become the speaker in this webinar. So I would like to share my knowledge in this webinar, and hopefully, all of us get the barakah, and also give us barakah to the development of the science in order for us to develop the drugs. Yeah. Okay. I think moderator already introduced me, through my brief CV. Yes, I work a lot in the herbal. I use metabolomics a lot, currently. So actually, the idea is that I would like to see the comprehensive view. When I start my study as a master student, I work a lot with the natural product, isolation of natural product, and also try to identify the bioactive compounds. And finally, I end up with the metabolomics. That's why now I use this technique very often, because I think this is a very effective and efficient method to study the natural product. Okay, let me share my screen, is possible? Okay, can you see my slide?
Yes, all clear, Prof. Alfie. Thank you.
Okay, thank you. Okay, the title of my presentation today, for this 1 hour, 1 hour presentation is the metabolomics in herbal medicine development. So before that, I would like to introduce my faculty. So this is the kuliah, in our university we call as kuliah. So this is like another name as the faculty. And then this is the kuliah of pharmacy. So we are located at the Kuantan City. So Kuantan City actually is in the east coast, of Semenanjung, Malaysia. While Kuala Lumpur is the west coast. So if you fly from Jakarta, you cannot fly directly to Kuantan. You need to have a transit at Kuala Lumpur, Kuala Lumpur International Airport, and after that you will continue your flight to Kuantan around 45 minutes. So this is the distance around 250 kilometers to 300 kilometers. Also possible by bus. For example, I think this is only 4 hours, traveling time. So because they have the highway, so it's not take a long time, as well by a car. Okay. So because we are very close to the beach, to the sea here, so we have a very beautiful view. We have beautiful beach here. So this is also one of the favorite touristic place, which is always visited by Bandar people, because Bandar people is from Kuala Lumpur and other part in Malaysia, especially in the Teluk Cempedak. Okay, so I come to our presentation. Okay, for the introduction, so the herbal medicine are extensively used in the pharmaceutical sector. So maybe you all know that the modern drugs actually is coming from this herbal medicine, and we learn a lot from nature. And this is like the source of database, database for the development of drugs. And then many company now they also focusing on the use of natural product without synthesizing, without modification of the structure. They just try to use from based on the traditional use, yeah. And then but then we have coming to the problem. Okay, the problem is the quality itself. Okay, so we are talking about the consistency, safety, and efficacy. We have a lack of standardization product, lack of the pharmacological knowledge, yeah. And in this case, we have a problem in toxicology, pharmacokinetic and pharmacokinetic data, as well as the dose response interaction studies. Yeah, so we are lack of this. Okay, because you know that in herbs we have thousands of metabolites, and then they work with the synergism, with antagonism. Yeah, so in order for us to understand, yeah, the bioactivity of the extract, of the plant extract, this is not the easy job. Okay, quality control, for example, if you are dealing with paracetamol, it is only one metabolite, so easy for us to control the quality. But herbs, we have thousands of metabolites there, that maybe the minor compounds also giving the significant bioactivity. So this is actually our bottleneck. Yeah? Okay, so this is what we always experience, yeah, in the lab, yeah, in our research lab, especially for the pharmacognosy student. Yeah, we have we did, for example, the bioassay guided fractionation. You have crude extract, and after that you fractionate, yeah, your crude extract, you get the fractions, and after that you have, and then each fraction you test again is which one is active, and you select the most active one. Okay, and then you try to purify them until you get the pure compound, and after that only you can identify the compounds. However, the problem is often that we get that the activity will be disappear after fractionation. Yeah, so we all understand why is it. Yeah, because there is the synergism effect, yeah, among the bioactive compounds in herbs, among the not even bioactive compounds, but among the metabolites in herbs. So if we isolate them individually, then we also have another problem, is missing the bioactivity. However, we need to understand how the metabolite affect each other. Yeah? So we don't know which metabolites having activity correlate to the activity when they are in the extract. But after we isolate them, then the effect is different. But we want to know, yeah, we want to know what is the effect of the metabolite when they are still intact to the extract. Okay, so that's actually also another difficulties, yeah, for the pharmacognosis. Yeah, in order to explain, yeah, the traditional use. Okay, and this is very important, yeah, to understand, to identify the compounds in the herbs in order for us to have the to proceed to the quality control, yeah, of the herbs. Yeah, yeah, maybe you know this figure, yeah? Blind man and an elephant. Yeah, so if you see them part of the elephant, yeah, maybe you will mislead. Yeah. Why? Because you didn't see this through the holistic way. Yeah, blind man, they saw the elephant through the reductionist way. Yeah, so you just check part of the body of elephant. Yeah. So at the end, they will conclude that this is elephant, but maybe they also mislead. Maybe this another things. It is not elephant. Yeah. Because why? Because they see this partly. Okay. Okay. If we come back to this flow chart, the same thing. Yeah? So if you see the herbs part of this only, then you will miss, yeah, you will miss a lot of things as well. Yeah? What happened actually, yeah, in the herbs. Okay, another difficulties. Yeah, see this one, yeah. So you have at the left side and the right side, yeah. Left side is our herbs. Okay, our herbs, we have complex metabolites, thousands of metabolites, and they work synergism and antagonism in order to give the effect, yeah, pharmacological effect, toxicological effect to human body. While in our human body as well, yeah, we have also a complex metabolite, yeah, which the pharmacological actions, the pharmacological pathway, yeah, is also very complex. The mechanism is very complex. It's not one, two. It can be million or billion of mechanism. So this also make the difficulties more difficulties. How can we explain, yeah, the pharmacological effect of the herbs? Herb is a complex system. Our body is also complex system, right? It means that, yeah, it means that we need a tools, yeah, a tools which can view this, yeah. You can view what happened in the herbs, yeah, and that also can view the human biological side, yeah. So then if we combine them, we hope that we can explain better, they can explain better, yeah, compared to what we have done before. Because what we have done before, we isolate the system. We isolate one compound here, for example, quercetin or glyphosate or glycol. So then we just test one of the mechanism. See, then we miss a lot of things, right? We already isolate some. One is this one, and we try to study only one mechanism. So then we become the blind man, yeah, blind man to see the elephant, right? So the idea is, how can we see all of this, and we see the impact also in the holistic way. So we hope that we can see, or our conclusion can be also holistic, yeah, not misleading. Okay, so then that's why, yeah, we come to the metabolomics. So we have done this one before a long time ago. I think we start in my lab actually is 2000 something, somewhere in 2005 or 2004. Yeah? So we are trying to understand this, yeah, trying in the plant system, in the human body, in animal system, yeah. So actually we are already use, yeah, other omics serology, for example, like genomics or proteomics, in order to see, yeah, the gene or protein involved, but we miss also metabolite, which is the end product, yeah, of this gene and protein. So metabolomics, by definition, is the comprehensive analysis of the whole metabolite, metabolite, yeah, under a given set of the condition, which is in the samples, yeah. So we are try our best to analyze as much as possible, yeah, the metabolite in the sample through multiple instruments. So we should use GCMS, LSMS, NMR, and etc. Yeah. So why? Because none of one single compound that can, yeah, analyze whole metabolite. Yeah. So the best one is to use the multi-platform, multi-platform of the analytical instrument. Okay. Okay. So then that's my introduction actually. Yeah. Why we go to metabolomics. So why I developed, try to develop different method, and what is the application on the in the pharmacognosy study. Yeah. So I focus on herbs. Yeah. I focus on medicinal herbs. So in my lab, yeah, I try to apply these three examples. Yeah. Actually, we have more examples, but this is what I focus nowadays currently. So I also work with also with the group, yeah, in UPM as well, in UM. So we are trying to work on this issue. Yeah. First, yeah, the example, the application, yeah, of this metabolomics is to in the analysis of the known bioactive compounds and screening of metabolites in the extract prior to isolation of unknown bioactive compounds. Yeah, so we are actually make a map. Yeah, so from the map we know, okay, this herb contain some of the bioactive compounds, which is can be known compound or unknown compounds. Can be the, if we isolate them, can be novel, or if we isolate them, it's not the novel compounds. Yeah. So before we isolate, actually we should make a map. Yeah, a map to in order for us to decide, yeah, which of the peak, yeah, or which of the spots, yeah, that should be isolated. Okay, there are so many cases, yeah, when we isolate the compounds which has been reported, yeah, or it's not novel, so we wasting of time. Yeah, so for example, one of my master's study, I just isolate a reported one, non-compounds, 2 years gone, yeah, because I isolate only the non-compounds. So we try to find the tools in order to map, yeah, to mapping, doing mapping for all the compounds. So our focus is only the compounds when we isolate them is become the novel compounds. Maybe I don't need to explain what is novel compounds. Yeah. So this is can be the new structure or the one which is with the new bioactivity, right? Secondly, is we need a very robust analytical tools in the quality control of herbs. And of course, this can be used for the process of optimization, yeah, based on the fingerprint of the metabolites. So now we are talking about fingerprinting. So fingerprinting meaning that we don't care what is metabolite inside. We just care the spectrum, just care the chromatogram. Yeah, this is like the thumbprint. Yeah? So as long as their pattern is in line, yeah, with the sample trend, then we can predict the quality of the herbs. Okay, I will explain later regarding this. Third is comprehensive bioactivity and toxicity evaluation of the herbal products. Okay. Yeah. So now, many cases, yeah, we test, yeah, our drugs, we test our extract, we test our isolated compounds, and yet what we analyze from the animal is also one metabolite, only glucose or creatine or creatinine, or yeah, and etc. So we only based on one metabolite or two metabolite, three metabolite. But however, yeah, in our body, in our blood, there are thousands of metabolites, yeah, which that can be influenced, affected by the metabolite of our samples. Okay, so it should be fair, right? Yeah, it should be fair that when you test your drugs, test your new samples, your new drugs, and test your samples, should not check only one target compound. That maybe this is your hypothesis, which may wrong. Yeah, so why not we just see everything? Yeah, so we check the effect on the serum, on the blood or urine of our animal, or maybe through the clinical trial. This one then should be analyzed through the comprehensive way as well. Okay. Yeah. So I think this is very technical. So I just explain briefly. Yeah. So actually, if you have samples and then you check the metabolite and you check the activity here, and then after that you have the statistical method, and then after that we can identify the bioactive compounds based on the result of the multiple data analysis, and how we identify is based on the first, based on the database. Secondly, based on then also through the NMR. For example, we use the 2D NMR, LSMS. We have fragmentation of Msms. Yeah, so we should also validate, yeah, our finding. Okay, and then by these wings also we can also try to find the new biomarkers. Yeah, try to find the biomarkers. For example, yeah, we develop animal become diabetes or become cancer. So, but our biomarkers, yeah, can be reported by, but the problem is that the only biomarkers may be another biomarkers that we never knew. Yeah. So in order for us to get to find a new biomarkers for the disease, then this metabolomics is also can be applied. Yeah. So if we combine these two data, then we hope that we can get the mode of actions. Yeah, and then we also can get the identified of the compounds, and at the same time we get the valid calibration model that can be used for the quality control of the herbs. Yeah, so this is the whole story of the study. Yeah. Okay, I think because I have only the short time, so as you see that now we have thousands of metabolite. So if you have 1,000 metabolite, meaning that we have many variables. So if you have so many variables, then we have a risk, the error, yeah, especially the type 1 error. So how we deal with that? Then we need to have the latent variables. Then we have kind of projection method that used by multivariate data analysis. Okay, so actually this is very technical. Yeah. So then, yeah, then by the new calibration model, the new calibration model can predict the bioactivity. Yeah. So for example, if you have herbs, okay, and many advertisement, they said, they claim, okay, these herbs have this can treat this disease, another disease, thousands of disease, right? Actually, maybe not correct. Yeah. Okay, let's say that we already have the use a certain extract in our lab or in our production site. Yeah. The problem of herbs is that the quality can be varied time by time. Okay. Even if you, for example, produce only one compounds, yeah, the quality of the samples is also can be varied time by time. Now, can you imagine we have thousands of metabolites there? So how can we control the quality? Yeah, then we need to have the calibration model, which already involve, yeah, consists of the thousand of metabolite, thousand of variables. So if we have this regression model, yeah, then if we have a new samples, then we can predict the bioactivity without, yeah, without we retest the bioactivity. Yeah. In other words, we can predict the bioactivity of the new batch of the herbs. Yeah. Only use a simple analytical instrument, for example, FTIR. Yeah. So without we retest using the in vitro, yeah, because it's also very expensive, yeah, then we will able to know the quality of our herbs in terms of the bioactivity. Okay, and at the same time we also will be get the result that our sample is belongs to which quality or belongs to which samples. Yeah, so very important in herbs. Yeah, because we are dealing with activity and we are dealing with the many of the quality of the samples. So we need to be sure that our sample is not the one which is outliers. Yeah, should be in the standards range. Yeah. And then for the optimization process, yeah, then we can use this model in order for us to optimize our product. Yeah. Okay, so I will come to the study case. Yeah. Study case number 1. Yeah. Actually, this one is we try to profile the metabolite, yeah, of the Clinacanthus nutans. This is the other word. This is the snake race, yeah, snake race, bali gajah, or in Indonesia we call it dandang gendis, yeah, dandang gendis. So okay, when we apply this method, so you have the plant sample with different quality, right? Plant sample with the different quality. So first, yeah, we should proceed with the fingerprinting. Okay, we should proceed with fingerprinting with the multi-platform, yeah, multi-platform instrument using LSMS, UCMS, NMR, yeah. And if you want to have fingerprinting, then you use the FTIR or a TLC. Yeah, TLC is also quite interesting as well. Yeah? And then and another wings. Yeah, so you have this set of sample with different quality. Yeah, then you also test on the in vivo, yeah, test or in vitro. Yeah. So what the meaning of this different quality? Yeah, different quality is different quality due to the different parameters to produce the extract. Yeah, can be the different temperature, yeah, or can be different what you call this air velocity during drying, or different solvent, yeah, or different place grow, yeah. So they have different quality, yeah. So because we have different quality of samples, yeah, and then also we test through the for the chemical profiling, and we will get the data of the bioactivity, yeah. So then we can combine them together of this data, and then using the multivariate analysis, then we can identify the bioactive compounds. Meaning that after that we choose the multivariate data analysis can give us the idea which peak, which peaks are related to the activity. So the multivariate data analysis can pinpoint this. The MDA can pinpoint the possible peaks that may contribute to the activity. And later on, the peak itself, we need to also identify the peaks is from which metabolite. So, for example, you have the peak in NMR. So in NMR, you have the splitting pattern. You need to check the chemical shift. You need to check also as well the coupling constant, and etc. And you need to see the SMBC, COSY, etc. And then after that, only you can identify the compounds, right? And only focus on the reported one. The one which is unreported or doesn't have database, then you cannot identify. How you identify? You need to isolate them, purify them, and after that only you can identify. All right. So I'm talking here is about the one which is already reported before. The one which is not reported before, then we should continue to the isolation. Okay. So meaning that we will isolate only the one which is new, and not the one which is already reported with their reported bioactivity. Okay. And then because we have already the model, calibration model, then we can make also a calibration, a valid calibration to predict the quality of the plant samples. All right. Based on the data that we input, give that we key in to the software. That if you have a new set of sample of new set of the profile, then our software then can also predict the activity of the new set of the samples. All right. So this is the one story. Another story is the bioactivity test itself. Okay. So for example, you are doing the animal test, using rat, for example. If you use rat, then you analyze whole metabolite in serum or in urine and other biofluids samples. Okay, when you already analyze, is using also SMS, for example, or GSMS. So then all the data you combine them, and then you process through this multiple data analysis. We already grouping them. This is the diabetic, this is the healthy, etc. So based on that, then we can identify why this animal is diabetic compared to the healthy. Which metabolite is higher? In which metabolite is suppressed? So then in this case, then we can have the biomarkers. Okay. So come back to my complete study case here, study case 1. So I only focus on this. Okay. I focus on the chemical profile, and I use the in vitro activity, in order for me to know the active compounds. All right. So this Clinacanthus, actually is traditionally has been reported as the to treat the inflammation, skin rashes, snake bites, allergic reaction, diuretic, and diabetes. So it has been also reported. So we try to find out what is the possible bioactive compounds there. So then we use the LCMS, we use the GCMS, and also we use NMR. Okay, from LCMS, yeah, we found okay, at the top one we call as the score plot, the bottom one we call as loading plot. The score plot, we found the activity on the non-polar compounds. Yeah, this is hexane compounds. So some of the people, they said, okay, hexane, I just remove it, because we don't care, because so many fatty acid there. Yeah, okay, but remember, there may be some of bioactive compounds also there. So what we found here is the one is activity higher. The most potent is the hexane. Okay, and then we test all the assay, and then and after that we analyze each of the sample using the LCMS. Yeah, and then what we get is the loading plot. The loading plot at the bottom figure. Okay, what the meaning of this plot? Yeah, so you have the green plot, the green spot, and the blue spot, and they have 3/4 here, 4/4, sorry. Yeah, so they are divided by the new latence variables, and we call as the PLS component 1 and PLS component 2. Yeah, so the green one actually is our peak at LCMS. So there is based on the mass, yeah, the ion ampere Z, yeah, and then at a certain rotation time, of course, yeah, and then the blue one actually is our the bioactivity. So in this case, we use at the time is the inhibition of the alpha glucosidase. Okay. So the value is the NAC50. So we divide them in order to make them equal, yeah, to the value of density of the of our sample. Okay. So we got this 1 per AC50. So meaning that if they are, the closer they are to the spot, so the more active they are. So the more significant they are to the activity. So this is the way how we identify, how we pinpoint, how MDA pinpoint the bioactive spot. Yeah. Okay, you can see here we have so many spots. Yeah, so many spots, and then we are interesting only which this part. There are actually a number here that we can call. Okay, this one is not important. This one is important. This one is induced enzyme. This one is can inhibit enzyme. So there's a range there. I will not explain here. Yeah, there are so many parameters in the MDA that we can say. Okay, in this range is the important peak. Okay, so then among these so many peak, we try to find the database and LCMS or SMMS, and then we get the similarity index. We get also the mass error. Yeah. So we just try to find from database, and we get only 4. Yeah, which is fit. Yeah. So we got this is terpene compounds. So we try to also to see the Msms fragmentation. Yeah. This is using the positive mode. Yeah. So we have tried to explain what is the fragment is. Yeah. So we need to explain. Yeah. We need to explain. Actually, I don't need to explain one by one. This is actually the MS mass. Yeah. Fragmentation is also quite long story as well. Step by step, we try to check. Yeah, although we database we know, but we also need to know to explain as well. Yeah, and this is like if you find the NMR or the peak, yeah, valine, for example, how the split, how the coupling constant, what is there. So the same as mass, you need to know. Although the database say this is similarity index is more than 90%, the MS error is almost 0. So, but we need to when we publish, we need to also give the information, yeah, to the journal, to the articles. So this is what yeah, this fragment is belongs to which ion, yeah. Okay, okay. So that's the LCMS. And then what about the GCMS? Yeah, what about the GCMS? So okay, I skip the score plot. I have the logic plot. So everything which is in the one side the same as the bioactivity, we consider them as the bioactive peaks. Yeah? This is actually we are talking about peaks. Yeah, peaks. Yeah? So the peaks with the certain rotation time. Okay, and then from the rotation time, then we try to find that peak is belongs to which metabolite. So then we check the fragmentation pattern, check the database using the NIST, and then after that we found, okay, this compound should be certain compounds. Okay, so from here we found out that palmitic acid, because they are the same site as the bioactivity, palmitic acid, phytol, number 4 is hexadecanoic acid, 6 monopalmitin, yeah, and 8 stimactin, yeah, and etc. Yeah, all this one is related to the activity. Yeah, while the one which is at the bottom, they are not in the positive correlation, yeah, to the bioactivity, which is like sucrose, yeah, and then also maltose or sugars, yeah, and also glucose not there, and 9 is gluconic acid. They are not have positive correlation, yeah, to the bioactivity. Okay, actually, yeah, this one is only the one which which is already selected. You see here we have the bar here, yeah, meaning that the one which is have significant impact, yeah, to the activity. We also have another variables or metabolites which is not reported here, is also can be identified, but they don't have any impact, yeah, to the activity. All right. So if you see the GCMS, you will see this is probability. So all probability are not above 90% is can be this as the compounds that we think this can be the right tentative metabolites. Yeah, this is only tentative metabolites only. Yeah. Okay, and then after you find out, okay, that compounds, yeah, then you try in the lab. Yeah, you try. Okay, I have palmitic acid, phytol, monopalmitin. You just try, and then try in the lab and check is it really active or not. When we found out this is active, it's active, yeah. So what? So it's in line with our metabolomic reports. Okay, actually, I have another story. Okay, I think I didn't put here. It's also interesting. Yeah? Sometimes we found that this is not in line with our metabolomics. When in metabolomics, they said this is active, but when we check, it's not active at all. Okay, I have some examples. This is very interesting. So, of course, yeah, we already assume that our identification procedure is correct. We have tried all this 2D, our LCMS calibrated well, validated well, everything is everything correct. But still, yeah, it's not in line with our metabolomics data. So I have tried in one table. I forgot the slide. So what we do is that we spike. So we spike that compounds to the extract, and we clearly found that when we spike them, the metabolite, it can improve the activity. Yeah, it can improve a lot activity, meaning that compounds they are working synergistic way with another metabolite. So they need the other metabolite in extract in order for them to be active. Okay. So if that one is isolated or in one single compound, they will not active. So they need other compound in the extract in order to give them activity. So this is very interesting part. Yeah, this we understand that's now we understand that the synergism effect in the herbs as well as antagonism effect. Yeah, and then it's also when we apply to toxicity, so also important, right? Now we know, okay, this compound for the bioactivity, another compound for reduce the toxicity. Yeah, so we can apply this one as well to our toxicity study later on. Yeah, so when we want to develop our herbal product, so we can enrich the one which is have positive relation to the activity, and we can reduce the toxic compounds possible like that as well. So so we can develop our product, yeah, in the future like that. Okay. Okay, this is only to confirm, yeah. So using the docking study, we have the molecular interaction, yeah. So we have with the hydrogen bonding and or other interacting residues. We check one by one as well. So accordingly, this is in line with our metabolomics data. Although we know that docking is only one ligand to the one protein, yeah, one side. So we also miss another part, which is may two compounds with one side, with one active site, or three compounds extra. We never know, but at least we can see the picture of the one ligand to one active site, or one ligand to one protein. Okay, so this is just to give the supporting data. That's what we predict in metabolomics can be true. Yeah, so we just metabolomics only prediction. Okay. Okay, this is the superimposed 3D, and we saw them clearly that there are the interaction, yeah, between the ligand and protein. Yeah. Okay, this is what I mean with the quality control. Yeah. I mean with the quality control, and you can use this after AR. So just use the simple method, yeah, simple method, and very cheap and very fast. Can we use that one for our fingerprinting or quality control? Our result said yes. So we have actually, I already try, yeah. My PhD student was already graduated in 2018, already published, yeah, in the vibrational spectroscopy. Okay. And we found out, yeah, of course, different quality of sample, they have different spectrum of FAR. Yeah, they are based on the wave number. Okay, so of course, if you see loading plot, there is the peak, yeah, in a certain wave number related activity, and this is the most important part, yeah, of the study, that this predicted IC50 should be almost the same as the observed IC50. So when we test, yeah, the bioactivity of our sample, of our extract, should be, yeah, direct actual result, yeah, of the bioactivity should be almost or similar to the predicted one which resulted from this model. Okay, so this one is the most important part. If the R square is less than 90%, then we reject the model. So we only accept the model which has the R square be above 0.9, meaning that the predicted IC50 and observed IC50, they are connected, they are in line. Yeah. Okay, so this is important for the quality control of the herbals. Another thing, one people ask me regarding, can we use this one to control the herbs in whole country or whole area? No, I think this one is for only our in-house quality control. Because why? You know that if you have this standard curve, meaning that you can believe the data of the sample which has been known, the parameter has been known, has been fixed. So if you have for example herbal from other region, then it is not valid anymore. So you only focus on your in-house product. Okay. So the aim is only for the quality control of your own product. Yeah. Okay. Another one, yeah. Study case number 2, yeah. So metabolic profiling of animal models. So we try to establish a rat and zebrafish. So we also use zebrafish now, yeah. So for toxicity, we also use zebrafish. For bioactivity, yeah. Also we use zebrafish now, yeah. So then, okay, we try with rat. Yeah, this is also important picture. Yeah. So we have tried before, yeah, to induce the animal become diabetic. So this is actually is we try to make this type 2 diabetic, meaning that the insulin level is not dropped, yeah, but the glucose is very high, yeah. So then what we get is, okay, the normal one after weeks 18, yeah, still the same as before, but you see the diabetic, the increase almost 4 times, yeah, and even after week 18. But the one which is with metformin and our drug with our sample, yeah, it can normalize, yeah, they can normalize, yeah, almost similar to the normal, to the healthy animal. You see this statistic, okay? So we only focus only one metabolite, which was a glucose. Okay, that's our conclusion. That our herbs can normalize the glucose level. The question is, what about another metabolites, which maybe we never test before? The problem is it possible to test one by one whole metabolites, 1,000 metabolites? That is very expensive, right? So then we try metabolomics. So we just analyze, yeah, using NMR, because in NMR there are well known. Yeah. So all this peak has been confirmed by 2D NMR. Yeah, I don't need to explain here. So why we call it the, for example, lactate? Why this is alanine? This is not like only by a proton NMR. We have 2D NMR as well, right? So we already identified all this one, yeah, all the possible peak. We try to find and identify. We have database and check. Yeah, we have this J-resolve and then check. Is it correct or not? Yeah. So what we get, yeah, we do the here using the discriminant analysis, meaning that we already discriminate them. Okay. So no, yeah, the result is that our herbs cannot normalize the blood profile and also the serum profile. Not, yeah. So glucose, yes, but not the other profile. You see here the normal one, and this one is the healthy one. Yeah. Okay. So this, oh, sorry, this is diabetic. This is the healthy, and this is the metformin, and this is our herbs. Yeah, it's not overlay. Even metformin, this is gone. Yeah. Yeah. So if you see the one parts, which is glucose, you can have different conclusion, right? Here metformin and our herbs, can normalize. Here, no. Okay, both serum and urine. So that's why I really like this one, yeah, because then we can see the whole picture. Okay, yeah. So when we check here, when we check one by one, the metabolite which has been identified here, yes, glucose, yes, can be normalized. Choline, no. Even choline, um, even choline, they can also shift the metabolite. Our herbs can normalize glucose, ethane, yeah, taurine, hydroxybutyrate, yeah, but not valid, but not ethanol, but not lactate, not glutamate, not glutamine. Okay, in some case, choline, for example, in STZ, in diabetic, the same as normal, but now we shift them to be abnormal. Okay, in urine, urine, citrate cannot be normalized. Glucose, yes, can be. Urea cannot. Okay, but others can be normalized. If you see the picture of metformin, you can see worse. Yeah. So this is what I only find in MR. I'll have another LSMS. You will find more metabolites because LSMS is more sensitive. Okay. Yeah. So that's for the pharmacology. Yeah. What about toxicity? Yes, we also can apply the same things. Yeah. So we can apply the same thing. So now we can see before LD50. You can want to see is there any change before that? You can check one by one like this. Okay. And then here, yeah. Now the problem is in vivo. So in vivo actually is we use rat. But when we have so many plants, we have so many herbs, it's possible to have many herbs, many animals. No, you will not be able to get even the ethic, yeah, approval. Yeah, so that's why for different sample I use in vitro. Yeah, but why? How can we have this in vivo then? If we don't have in vivo, so then we miss, yeah, whole mechanism. So then come to the idea, why not we use zebrafish? I found in many articles that people develop zebrafish already. Okay, so the beauty of zebrafish, I didn't see the beauty of zebrafish is the larvae, yeah, the larvae parts you can put in the well, an anti-6 well, you can put them. So one well considered as one in vivo condition, right? It's not in vitro anymore. So we just use 96 well. One well can be put larvae. It's just very small, this larvae, right? Very small. So we have tried to establish diabetes. Yeah, so we are happy that of course we can get also the model. And if you compare, again, the same story as rat. If you compare, if you use one parameter, you find that okay, your herbs can normalize the glucose, but what others? No, they cannot normalize. Still cannot. Yeah. So this is also using LSMS, using zebrafish. Yeah. So later on, but I still use adult. But later on, we will develop with larvae. Yeah. In order to have a better understanding, yeah, of our herbs. Okay, yeah. Remember, I only focus on only one herbs. In traditional use, they use a lot of herbs. It's another things, yeah. So be happy, yeah. We have so many problems. Still have some sample, okay, yeah. So then we tried how we explain. So the only thing we can do is by zebrafish. I think nowadays I cannot think other way. Okay. So for conclusion, yeah. So this is our conventional approach. Okay. First, we try sample, we try in vitro tests. We have glucose up, for example, glucose uptake, DP4, etc. And after that, we go to testing to rat, right? After everything is positive or some of positive, we go to testing on rat, and after that only we go to clinical trial. Of course, we need to do toxicity, but our future approach now different. Once we get the zebrafish and we get the optimized condition of our drugs, our same extract, now we just screen using zebrafish toxicity and also the bioactivity, and we test the metabolomics, we test the proteomics, we test the genomics. Okay, then we get the mechanism there already. In order for us to confirm, then we go to in vitro. So in vitro is only to confirm, not to screen. In vitro, not to screening anymore. Yeah. So we just to confirm that what we get is correct. Yeah. Then after that only we go to testing on rat or mice. The optimized product, that this is already the product, and only we go to clinical trial. So we hope that we don't want, we will not waste our time. Yeah. Sometimes in vitro active, in vivo no. Or in vitro not active, in vivo yes. Yeah. I experienced that many times. So now our own future approach is this one. So in IUM, we are collaborate with the Faculty of Medicine, Faculty of Science. Some of them, they work with the cancer. Some of them with the one healing. I work with diabetes. And so we divide the job. So, okay, in the future, we want to have many models in IUM. And then if we want to develop the drugs, so we just use their zebrafish. Okay.
Prof. Alfie, excuse me, you have
Yes.
10 minutes left.
10 minutes.
Yeah, thank you.
Okay. So, so this is the our future study, our another application. Okay. So you have medicinal herbs complexity, human complexity. Okay. So if you have this, yeah, of course, before we are to study on the animal, in this case is zebrafish, right? So in medicinal herbs, we have many of metabolites. The next step is to understand the mode of action of each of metabolite on the mechanism, each mechanism here. So that's our dream, yeah. Of course, difficult, yeah, but we just try. I think this is, then we will have the holistic picture now, okay? Using the X variable, this metabolite become X variable. This one become our Y variable. The multivariate analysis we use is the partial least square, yeah. So hopefully it can give our better understanding on our herbs in the future, and also not only one mechanism but many mechanisms in our animal model. Okay. Now, so another thing is our approach is not bioassay guided fractionation but metabolomics guided fractionation. In order for us to isolate, better for us to do the mapping first. So we will have this how we select the sample or the fraction is based on the one which has been mapped, yeah, by the metabolomics techniques. Okay, so the conclusion, yeah, so this method open the possibility to faster the identification of the novel bio and chemical markers. So chemical markers in herbs, the biomarkers in the animal, yeah, and then it can also have produce the calibration, yeah, validated calibration model in order for us to have the quality control in house, yeah, and we also can have comprehensive study on mode of actions and also have toxicity sample that we can be detected earlier. So yeah, all of this one is really good tools in order for us to get the product which is enriched with the bioactive compounds, and possibly we can remove the toxic compounds. Yeah, this is our dream. So who knows? Yeah. Okay, that's all. Yeah, thank you very much, and let's collaborate. So I give back to the moderator.
Thank you very much, Professor Alfie. It was unnecessary for me to remind you of the timing. Yeah. I apologize for that. Once again, thank you for the talk. It was a very interesting talk, and I'm sure it has sparked some questions among the attendees, but please hold on to your question for now. We will save it for later, because now we will have the presentation from our second speaker, Assistant Professor Siti Hadijah Samsudin, in the development and commercialization of herbal medicines. Dr. Siti Hadijah Samsudin obtained her Bachelor in Science in Pharmacy from Liverpool John Moores University in United Kingdom. Afterwards, he worked for retail pharmacies as pharmacist as well as manager for about 10 years. And then she later worked as hospital pharmacist and manager as well. She then pursued her Master in Business Administration from University Utara Malaysia. Afterwards, she started her career in academia and later was awarded a PhD in Pharmacy Management from University Technology Mara, Shah Alam in 2010 for her study in Good Governance for Medicine. And then she joined IIUM and was appointed as the head of Department of Pharmacy Practice, Kuliah of Pharmacy, and head of Department for Regulatory, as well as registration for ECOP Manufacturing Plant, IIUM Holding since 2013. She was the former dean for Kuliah Pharmacy, IIUM until 2016. And now her current position is assistant professor at Department of Pharmacy, the head of regulatory at ECOP, and the head of quality assurance at ECOP. So many titles appointed to her. Assistant Professor Siti, this is quite interesting how you jumped or explored. I think explore is the better word for this, from community pharmacy to becoming academia, and then how you landed a position at ECOP, is a pharmaceutical industry. I'm really excited to listen to your talk, and now without further ado, the floor is yours.
Can you hear me?
Yeah.
All right. Thank you very much. So let me share my slide. Thank you, Dr. Alfie, for giving us the very interesting view about this metabolomic. But let me share my screen. Host only can share the screen. All participants. Okay.
Please, for the host,
All right.
can you appoint Dr. Siti as the host now, or co-host?
Okay, I try. Can you see anything?
Yeah, we can see your screen now.
All right. Let me go up. Okay. So just now I was told that my talk today will be on the commercializations of these herbal medicines. But I'm going to talk about standardization of raw materials of herbal medicine for commercializations, because well, I'm going to talk from the view of pharmacy and regulatory. Okay, so the talk that was given by Prof. Alfie just now was in line actually when it comes to the explanation of the needs of all the requirements for the registration of the products. Because like I think in Malaysia and the whole country as well, when we want to commercialize herbal medicines, we need to get it registered with whatever country that the products is going to sell, okay, or used by the populations. So let me just share with you all a little bit about the standardizations of raw materials of herbal medicines for commercializations. Okay, so well, maybe it is too wordy, but as a regulatory people, this is sometimes very crucial for us. Okay, I want to make it. So as we know that Malaysian rainforest acknowledged as the world's oldest rainforest, ranked 12 in the world for the biodiversity hotspots of Asia after India, China, and Indonesia. So I think Malaysia, okay, Malaysia actually we has estimated about 12,500 species of seed plants. Yeah, so All right, sorry, this is for the 12,500 species of seed plants is all over the world. And then among these, approximately we have about 1,200 species reported to have medicinal value. Okay, and WHO defined herbs as crude plant material covered different plant parts which exist as whole, fragmented, and powdered form. Okay, and then herbal consists of herbs, herbal preparation and mixture, or finished herbal products that contain active ingredients from plant parts, other plant materials, and for example, product made from plant materials such as outer bark of the cork, oak being used for stopper for bottles examples, and mixture of both herbal medicine. This is from Herbal Medicine Research Center. Okay, so we can see that actually herbal medicines is actually coming up now. It's one of the interesting part of the pharmaceuticals to be used, to be consumed by our patients. But of course, due to certain conditions, it is difficult for us to convince healthcare professionals in order to use these herbal medicines for clinical usage, because in Malaysia now, actually we have regulations that we call it herbal medicine, traditional herbal medicine with indication. So the one that just now what Dr. Alfie Khatib is telling us are very crucial in order for us to go for that direction, meaning to say that we want to register herbal product with indications. Okay, as you know that to register herbal product is not as crucial as or as typical as pharmaceutical products, where pharmaceutical products, they need to have a clinical trial before the product can be registered and used by the patients, but not for herbal medicines at the moment in Malaysia. So in order to ensure the quality, safety of herbal medicine, Malaysia actually has developed its own monograph with the vision of transforming them into a pharmacopoeia in the near future. So I think with the job that or the research that Dr. Alfie is doing, inshallah, Malaysia can go, can have their pharmacopoeia structure very easily, inshallah, in future. So the monograph contains specification for the identification of plants using single chemical entities known as marker compound or profiling technique analyzed with chromatographic methods. So the monograph document can strengthen value chain supply since it is applicable to raw material, extract, or herbal product, as well as monograph enhance the research and developments of the herbs. So this is the needs of our herbal leading towards. Okay. So as I told you just now, there's a lot of wordy in my presentations. So issues pertaining to raw materials are considered crucial as they are the first step in development of herbal medicines. So for example, the source of plant, whether they are found in wild or cultivated in plant field, will affect the characteristic of the raw material, because it's now Dr. Alfie said that in a plant, there are lots of variables. There are lots of materials that we don't know which one is good for which one has got the therapeutic value. So other important factors are related to biodiversity, conservation, and ecosystem sustainability, as well as maintaining the genetic diversity of the raw materials. So factors that may affect sustainability include development or resettlement of forest land, over-harvesting or populations. So actually these are the issue of us when we want to get the source of the raw materials. We want to have the standardized extract, the extract that we want to use for our productions, for our whatever products that we are going to produce will require a standardization extract. So to overcome some of this problem, the raw material sourcing, the GACP guideline, a document which guide raw materials growers and collectors, is published actually, and this ensure that when raw materials are collected, they are correctly identified, meet quality assurance specification, not contaminated with unsusceptible levels of substance, and harvested during optimal harvest condition with focus on environmental sustainability, etc., etc. Okay, so actually commercialization of herbal medicine in Malaysia, it is interesting to note that with a precise understanding of effective dose, safety, and mechanism of action, herbal products have been proven to help in promoting health. So the various studies make several noteworthy contributions to the health benefit of these medicinal plants. In this modern era, interest and demand from plant sources commercialized product is increasing actually. If you can see that there are, especially in Europe country, US, they use a lot of herbal medicines, but we from Asia, sometimes it is difficult for us to make our product commercialized over there. They will, for example, like they will use our products and do the commercializations. Okay, so it is very, so in future, maybe we hope to do this. Yeah? So the improvement of herbal product quality also can be boosted up as Research and Development Institute, Agriculture R&D institution in Malaysia have been established to work toward this practical solution to solve all these problems. Yeah? Okay. So I just want to share with you, actually, this is from IMR. So these are the what is happening actually to the raw materials, or when we want to get the products to be commercialized. So at first, selection of the herbal plants need to make sure that all the where will be the raw material sourcing, etc., etc. But when you look at all these things, this is the from the regulatory of Malaysia, NPRA, National Pharmaceutical Regulatory Agency. These are the documents or the items that is required when we want to register our products. Number one, the identification of the products, and number two, purity and safety of the products. And as I can see that all these things, all this, one of the method of doing it just now was explained by Dr. Alfie. Okay, if you see here, for examples, all right, for examples, these are the data, and then data specification by NPRA. If we want to produce our monograph, all these things are important. Okay, all these informations from the herbal are important in order for us to produce our monograph. Okay. And then, as everyone know, because I think most of the students and researcher, you understand about these herbals very well. So actually, I'm just sharing with you just a little bit of reminder for you all and for myself as well. What are important when we are doing the standardization of herbal drugs from the botanicals. These are the things that we are going to see. Chemicals, just now, chemicals, chromatographic technique, heavy metals, pesticide residue, and mycotoxics. These are the things that we are going to get from the herbals or to study. And then the physicals like moisture content, extractive value, ash value, and the biological also like microbial contamination, pharmacology evaluation, and toxicology studies. Actually, all these are the documents required when we want to register our products for commercializations. So if you look at here, as a pharmacy students, actually that's why from year 1 until I think year 3, they will do a lot of study, or they will learn a lot of things on basic sciences to make sure that they can do all this later when they practice as a pharmacist, especially if they want to work in the industry. Because I am sharing this from my experience. I never had experience working in the industry before, because I was working as was introduced as now in community pharmacy, where we use all the finished products. Hospitals also, we use all the finished products. But in hospitals, rarely we see herbal medicines. But in the pharmacy nowadays, there are lots of herbal medicines. But as a pharmacist, how are we going to choose which herbal medicines is the best to be used, and the authenticity, the safety, the safety, the efficacy of the medicines? Difficult for us to say which one is good, because there is not much clinical trial done for herbal medicine actually. But at least all this information can help us or can guide us to practice as a pharmacy for herbal, or to be a herbalist in future. Okay, so these are the definition of standardization. Maybe just a refresher. Standardization means confirmation of its identity and determination of its quality and purity. So that is the definitions or what is meant by standardization of herbal. So the quality control standard of various medicinal plants used in indigenous systems of medicine are significant nowadays in view of commercialization of formulation based on medicinal plant. Okay, so meaning to say that last time we just take the plant from our forest, but not only that, Dr. Hafi, we have to actually do the little bit of standardization. This is the problems with us now, where it is quite difficult actually to produce, or to produce, not to have, kalau purchase maybe we can get, to produce a raw materials with the standardization extract. So that one we have to leave it to those people who are from the agriculture side. Okay, so what are the herbs standardized? How and what are herbs standardized? A standardized herbs extract is an herb extract that has one or more components present in a specific guaranteed amount, usually expressed as a percentage. So how are we going to do all this? Maybe with the help of Dr. Alfie methodology, we can do this. The intention behind the standardization of herb is to guarantee that consumer is getting a product in which the chemistry is consistent from batch to batch. Okay, so that is our, as a pharmacist or as a healthcare professionals, that is our concern, to get a consistent the chemistry is consistent from batch to batch. And then these are the drugs or formulation are expected to exert a desired biological activity at particular concentration of their chemical constituents. So the overall aim of drug standardization is to ensure the quality, efficacy, and uniformity of the products in terms of their chemical and biological properties across the manufacturers. That is our aim actually, for the needs of the standardizations, formulations, of course, from the raw materials, the extract of the raw materials. Okay, so these are the difference between extract and there is no difference actually. There is no necessary difference between whole herbs and standardized extract. So any extract, including a whole herbs or broad spectrum extract, can have constituents quantified and thus be standardized. So some standardized extract, however, use process and or solvent to isolate the phytochemicals. So that if you mean in Kuliah of Pharmacy, we leave these phytochemicals kind of study to those people at Department of Pharmaceutical Chemistry. Okay. They, I think, is there our students, your students here, is there any, the participants here are all students from undergraduate or postgraduates?
Mainly up from undergraduates. So they are.
Thank you very much. Okay, thank you very much. So those are the things that usually will happen at year 1 and year 2. So they will learn how to do this extraction and understand the needs of these herbal products. So what are the advantages? The advantages of standardization. Advantages of standardization, like rationalize different varieties of products, decrease the volume of product in the store and also the manufacturer cost, improve the management and the design of the products, speed up the management of orders, facilitate the exportations and marketing of products. These are very important because without the standardized extract, sometimes we cannot sell the products. Okay, sorry. And simplify purchase management. So easy for us lah. When we want to produce a product, we try to get the standardized extract so that when we want to label the constituents of the product, it is there. Otherwise, difficult. Okay, that's why when we want to register products without the standardized extract for pharma products, it is not easy to register the products. Okay. And then these are the, I just share with you the WHO guideline for quality standardizations for quality standardized herbal formulation. A, quality control of crude drugs material, plant preparation and finished product is required. The stability assessment and shelf life are also required. Safety assessment, documentation of safety based on experience or toxicological studies are required. An assessment of efficacy by ethno-medical information and biological activities evaluation are also required. So, okay. So all these informations are required when we want to have the document, when we do product registration. And so the bioactive extract should be standardized on basis of active principles or major compound along with chromatographic fingerprints. Just now, I think Dr. Alfie already shared with us the detail of how to do all these things. Okay, it is not my expertise to explain. Luckily, Dr. Alfie already give the talks earlier, so easy for me just to highlight. Okay, so the standardization of crude drugs material include the following steps. Number 1, authentication, stage of collection, part of the plants collected, regional status, botanical identity, like phytomorphology, microscopical and histology analysis, taxonomical identity, all are required. So that's why when we purchase a raw materials, we need to have all this information from the suppliers. And then we also need to have the forage matter. For example, herbs collected should be free from soil, insect parts, or animal, etc. This is also the information that will be given by the supplier. Organoleptic evaluation, sensory character, taste, appearance, odor, whatsoever. Tissue of diagnostic importance present in the drug powder, for example. H values, volatile matters, and moisture content. More. Chromatographic and spectroscopic evaluations are very important as well. Determination of heavy metals, example, cadmium, lead, arsenic. Pesticide residue, for example, WHO and Food and Agriculture Organization set limits of pesticide which are usually present in the herbs. So we have to know what is the content. So these pesticides are mixed with the herbs during the time of the cultivations. So mainly pesticides like DDT, BHC, Taxophene, Aldrin cause serious side effect in human being if the crude drugs are mixed with this agent. So we need to have the information from the supplier on this pesticide residue. Okay, and then microbial contamination. Usually, medicinal plant contain bacteria and molds coming from soil and atmosphere. So the analysis of limit of E. coli and molds easily throw lights towards the harvesting and production practices. So usually, when you want to purchase, these are the required information as well. And radioactive contaminations. Okay. So examples, these are the limits for microbial contaminations. When we want to use that particular raw materials as our products, as our ingredients for our products, we have to make sure that the finish, for example, these are the microorganisms for the finished product and for the raw materials expected. Okay. And in order to obtain quality-oriented herbal product, care should be taken right from the proper identification of plant, seasons, and area of collection, extraction, isolation, and verification process. It's not easy, yeah? So chemical and instrumental analysis are routinely used for analyzing synthetic drugs to confirm its authenticity. So the authenticity is the problem for us, actually, when we want to produce a raw materials, because we cannot get the information from the farmers on all these the documents from the farmers to ensure the authenticity of the products. In the case of herbal drugs, however, the scene is different, especially for polyherbal formulation, as there is no chemical or analytical methods available. Therefore, biological screening method can be adopted for routine checkup of herbal drugs and formulations. In the case of herbal drugs, the quality of raw materials and products can be furnished by regular pharmacognostic identification and phytochemical analysis. The herbal formulations in general can be standardized systematically as to formulate the medicament using raw materials collected from different localities and a comparative chemical efficacy of different batches of formulation are to be observed. So, and then the preparation with better clinical efficacy are to be selected, meaning to say that if we want to produce a herbal medicines, we have to make sure that the information about the chemical constituent, the biological constituents of these herbals, we know from A to Z, from the beginning of the process of getting the raw materials. Okay, so the stability parameters for herbal formulation, which includes physical parameters, chemical parameters, and microbiological parameters are important. For example, physical parameters include color, appearance, odor, clarity, viscosity, moisture content, pH, disintegration time, friability, hardness, flowability, all these are very important. And chemical parameters include limit test, extractive value, chemical assay are also important, and chromatographic analysis, as explained by Dr. Alfie just now. And then these are the microbiological parameters, total viable content, total mole counts, etc., etc. And chemical composition of substance present in the formulation also produce several toxic or impure compounds during storage is undesirable conditions. So that's why we have to report all this first. When we want to register the product, all the documents with regards to all these things are very important for us to have. Okay, and again, This is a guideline set by WHO, like a reference to identity of the drugs, reference to the physiochemical character of the drugs, reference to the pharmacological parameters of the products, toxicity detail like heavy metals like cadmium, lead, arsenic, mercury, and pesticide residue are very important. And then how to calculate the maximum residue limit. This is how we do it actually, yeah. And then main daily intake of the drug and safety factors. So all this not as stringent as for pharmaceuticals, but we need to have the information in order for us to register the products. So for example, like nowadays, we have a lot of herbal Ayurvedic, because herbal Ayurvedic, they have got a complete monograph. In the modern herbal Ayurvedic monograph, the standardization parameters discussed in a very comprehensive way, actually. So according to the modern Ayurvedic monograph, the quality control protocols include the following: titles, synonyms, publication related to the plants, constituents present, analytical methods is available. And they also have the descriptive evaluation, descriptive of the plants or the drugs, phytomorphological, microscopical, everything is there. Actually, for Ayurvedic, they are very up to date with their products. Indian herbal, I think, call it. And then the physicochemical parameters like identity, physical and chemical identity, chromatographic fingerprints, H value, extractive value, moisture contents are there. Okay. And then these are the biological activity evaluations like bitterness values, astringency values, swelling factors, form index, and hemolytics index. Why I said just now the importance of this is that when we want to purchase the raw materials, for example, we refer to this Ayurvedic, we as the manufacturer, we can always go and buy the raw materials. If they have all this information, it's easy for us lah when we want to register the product, because all the documents are available. Okay. And then they also have the toxicological evaluation, pesticide residues, heavy metals, microbial contamination, etc., etc. And the presence of aflatoxin can be determined by chromatographic method using standard aflatoxin B1, B2, G1, G2 mixture. This aflatoxin is a product of microbial strain Aspergillus flavus. And I was made understand that this aflatoxin actually available in the raw materials. If it is available in the raw materials, it can be very dangerous for the products. Okay. And then they also inform us about the radioactive contaminants and therapeutic usage. So basically, this, and then they also have the classical therapeutic attributes like Rasna, Guna. These are the Ayurvedic terms. I also not very sure about what is Rasa, Rasna, Guna, Virya, Vipaka, and Karma. Classical formulation like doses, storage, because all this, it depends on the their country, how they define it. So quality of raw material can be tested according to the following lah. They have name of the drugs, part of the plants, area of collection. All these informations are very important when we want to purchase the raw materials, or when we want to produce the standardized extract of a raw materials. Okay. So actually for us, we have two duties. One is to produce a product. Another one is to produce a raw materials. So if you want to produce a raw materials, these are the things that is required. And then if you want to purchase a raw materials to produce our product, we also need to have all this information whenever we are buying the raw materials, so that it will be easy for us when we want to register the products, since all the documents are available. Okay. So here, just a share of information, analysis of raw materials from plant. So raw materials can be defined as starting materials or any intermediate which will be utilized for further processing, as I told you before. So before finished pharmaceutical dosage form are produced, the identity, purity, and quality of raw materials as per specification for impurities and other related substance present must be established with use of suitable test method. Okay, pharmacopoeia and formularies of various countries provide standardized test method for the most common and widely used materials in their monograph. So if our product already have if our raw materials already have the monograph of that products, will be easy for us. Okay, if not, then we have to produce that. So the stored drug samples are prone to attack by harmful mycotoxin for producing fungi just now. So detection of mycotoxin, aflatoxin B, acrylatoxin, all these mycotoxin is certainly a matter of great concern in stored drug of important medicinal plants. So that's why whenever we purchase the raw materials of herbal medicines, we have to make sure, we have to check all these things first. Okay? So I just want to share with you, I stopped first this presentation because I want to share the guideline on registration of natural product in Malaysia. Okay, I think I still have sometimes. I stop sharing for a while because I want to scanner. All right, just for a while. Okay. Okay. Sorry, I did not open my. Where is this just now? Okay. Okay, let me share screen. Okay, so I just want to share with you all about this. We in Malaysia, we do have this guideline on registration of natural products. So actually, herbal medicines falls under this category. It's very easy. You can just go to the drug registration guidance document, third edition, third revision July 2022. So this is the latest guideline to register the products in Malaysia. We do have all the general information and then detail. Can you see? Can you see my slide?
Yes, all clear.
All right. Okay, thank you. So if you look here, we have definition of traditional products, etc., etc. Exemption from product registration is also there. What are the things that we cannot register? For example, like we cannot register animals products. Preparation which are not allowed to be registered also available, and classification for specific active ingredients also they will tell us clearly. For example, like product containing cassia, senna, all these things, they will tell the details about it. I go back. Okay, and then we also have the information about general requirement for registration of natural products. And then what are the things that is required? The details. Okay. So actually later I can share with you all this and others my simple lecture notes and this guideline just for you all to see lah. All right. Okay. And then I go back to my presentations. Stop. Stop sharing. Okay. And then I go back to Can you see my slide?
Yes, we can see your slides.
All right. Okay. All right. So just now, I will share with you the guideline on registration of natural product because it's going to be lengthy if I were to explain here. I need another 1 hour. Okay. All right. So as a conclusions, okay. So as a conclusions, now I think the subject of herbal drug standardization is massively wide and deep. And then there is so much to know and so much seemingly contradictory theories on the subject of herbal medicine and its relationship with human physiology and mental functions. Because of when we are trying to, when we have a plant or herbs, we are trying to get one active ingredients from the herbs, but we don't know that actually maybe the products require all the ingredients in order for it to be or to have the therapeutic value. Okay, so I think with all this, maybe more study, or we need people like Dr. Alfie to tell us what, for example, like if we have herbals, maybe not that difficult. Like if you have a foods or fruits or whatsoever, so we don't know actually what is the best. Is it when we isolate the products that is the active ingredient? For example, like ginger, then we have gingerols. We have all sorts of ingredients inside there. Is it work if we isolate each of the products? That one not so sure. So for the purpose of research work on standardization of herbal formulations and nutraceuticals, a profound knowledge of the importance herb found examples in India and widely used in Ayurvedic formulation is of utmost importance. So maybe if possible, try not to isolate the ingredient, yeah, Dr. Alfie? Is it good to isolate or to keep the whole plant for a formulation? That one, I think we need to hear from others after this, from their perspective as well, because for us, we just want to get the products, get the raw materials or get the plants, and then we want to produce a product. But which one is good, whether isolations or the whole plant, that one we leave it to people that know about this better. So for example, like India and other countries can emerge as a major country and play their lead role in production of standardized, therapeutically effective Ayurvedic or herbal formulations. Maybe Indonesia, you are already very, I think we have a lot of your products actually in our market. Okay. But Malay traditional herbs also available. It's just that sometimes we need to help our cottage industry in order to make the products available internationals. Okay, so that is our duty lah. So there is a need to explore the medicinal important plants. This can be achieved only if the herbal products are evaluated and analyzed using sophisticated sophisticated technique, like what Dr. Alfie is telling us just now. Yeah, so technique of standardization such as UV visible, TLC, HPLC, all these things. Yeah? So with the informations and with the analysis that was done by those people like Dr. Alfie, I think, inshallah, in future, we can have a better, or we can have a lots of formulations or products from herbal medicines, from herbals. Okay. So these are the reference if you want to see for my talk just now. Actually, these are the references that I use. So thank you very much for listening to my talk. Okay.
Thank you very much, Dr. Siti. It was a very exciting talk about the commercialization, especially the standardization of the raw materials. We can relate more, especially for our undergraduate students, and because we have the herbal medicines industry here as our attendees, so maybe they can ask for questions later. Now we will go ahead and take some time for the questions and answer session. I would like to remind everyone that you can raise your hand or type your questions in the chat box, and both questions in Indonesian and English are welcome. We actually have 2 questions so far. Those are from Ibu Dinas. I will read out the first question. It is for Professor Alfie. The question is, as you mentioned, the holistic approach for herbal medicines, metabolites profiling, and bioactivity metabolite profiling is necessary for understanding the mechanism. My question is, which one is more important? Is it to investigate the herbal metabolite profiling or the biological study metabolite profiling, as well as necessary to do them simultaneously? Or we may just choose one to go first and then follow up with the second method later. As we know, sometimes it is difficult to go together in research. Thank you for the answer. So please, Professor Alfie.
Okay, thank you, Dr. Erna. So just say hello to Budinar. Budinar is, we are actually the old friend in Leiden University. Okay, regarding the questions, yes, I said that we need the holistic method. Okay, for both of this chemical profiling and biochemical profiling. Yeah. So which one is should be more important? So it is depending on the facility that we have. If we have the model for zebrafish, yeah, if we have model for zebrafish, then we can both them. Yeah? We can proceed to the both approach. Yeah, we test to the herbs and also we test also our animals. But as long as we don't have the zebrafish model, so this is impossible. Okay. Why? Because if we use rat, for example, rat is considered only for one sample only. Yeah, for example, ethanol extract or water extract only, or maximum 2 or 3. If you have already 2 sample, you require more than around 250 rats. So this is, I think, it's quite difficult as well to get the approval, ethical approval. Yeah. So, but in order for us to understand the mechanism of action of each of metabolite in herbs, then we need at least 30 sample. Yeah, yeah. By 30 sample is enough to understand the mechanism of action of each metabolite of our herbs. Yeah. Using the holistic method. Yeah, it means I mean the using the biochemical testing. Yeah, So if we don't have zebrafish, then we need to have one mechanism only. For example, using in vitro. Then we can use the metabolomics part for our herbs. Otherwise, you will have one sample herbs, and then you test on the animal using metabolomics. So if you use rat, yeah, so you can choose. You can test your. You can use the herbal metabolomics for your herbs, or you can use metabolomics for your rat, yeah, or animals, but only use one sample. So actually, the bottleneck here is zebrafish itself. So that's why we are working very hard, yeah, to get the model of zebrafish. Up to now, for example, in diabetic itself, it's quite difficult as well, yeah, to get the larvae, yeah, larvae state. Instead of analysis of glucose is also difficult because its larvae is very small. We cannot just analyze the blood, yeah. We need to, if larvae, we cannot analyze the blood only. We need to crush all the body. Then this is accurate, yeah. But if we use, for example, analyze the targeted gene, then possible. But this is also expensive. Yeah? So the thing is that the answer of the question of the webinar is depending on the facilities, right? If we have zebrafish, if we have this, then fine. We can evaluate this easily, right? Okay. Hopefully, it can satisfy you, webinar.
Thank you very much for the answer. Mr. Alfie, can I add you further question related to the first?
Okay.
Yeah. Instead of using the zebrafish, do you have an experience to the biological activity using the rat or the mice? Usually, it is a standard method to do the pharmacological study, and we use mostly the urine or the blood instead of kill them. Is it possible to analyze those samples to relate the activity with the metabolite profile of the herbs? Thank you.
Yeah, we can use rat or mice, but as I told you, we just use one sample or two sample only for one experiment. Then it means that we are explaining the activity of the extract, but not explaining the activity of each metabolite of the extract. So we are dealing with the meaning that we try to explain the activity of the extract. So the questions is how is the activity of each metabolite? So that's why if you have zebrafish, then we can explain this. Yeah, that's why this is limitation,
Yes.
right?
Yes, and I understand the point. Yes, so it depends on the study we want to investigate. Yeah. Okay. Thank you for the answer.
You're welcome.
Thank you, Budina, for the question. I think now we can move on to another question from Budina again to Dr. Siti. I will read out the question first. So during the standardization for crude drug material, how did you apply so far? And as we know that herbal raw materials might vary depending on the herbal source, how to minimize these issues? This is actually an answer from Dr. Siti in the chat box. The guideline is given by the WHO. To minimize this issue, we need to work with the growers, and we need to get the plant from the forest if it's from the natural origin. And that Dr. Siti agrees with the opinion from Ibu Dina on the variable. So do you want to add something more to this, Dr. Siti?
Yes, thank you. Thank you, Dr. Siti, for the explanation. I want to explain more about my question, because in Indonesia, usually for the commercial production, it is how to get the large amount of the simplicia. It is very difficult, especially with the standard one. And then for the to collect the wild plant, it is sometimes a bit tricky, because the standard, what is it? The standard, let's say the metabolite profile or the standard active compound, it is sometimes not fulfill the requirement. So in ECOP, usually how you deal with this issue? Thank you.
Okay, thank you very much, Ibu. Actually, in ICOP, what we do when we want to use the raw materials from herbals, we have to check that the raw materials have got all the requirement that the WHO already give us. Otherwise, at the moment, we won't produce it, because it's going to have difficulties for us to register the products. For example, actually in ICOP at the moment, we do not have any herbal medicine or traditional herbal products. We only have ointments. That ointments, actually we use eucalyptus oil. Eucalyptus oil is one of the herbals, plants, from the plants as well. So what we do is that we only get this eucalyptus oil from the source of materials that have got all the documentations that is required by our regulatory agency. See? So that is our approach now. So that's why, as I told you just now, I understand that it's not easy for us to, because when we want to produce raw materials, those are the things that we have to take into consideration. So I think nowadays we have to work with growers, and then we have to need to have a talk with all these agriculture people to ensure that they can provide us with the products, or they can help us to grow the herbals or products that we want according to the standards that is required. If they want the product to be in the pharmaceutical formulary. Okay.
Thank you for the response, Dr. Siti.
All right.
Is there any more follow-up question, Ibu Dinar?
Actually, for us, ICOP, not only standardized extract that is required, we also have another difficulties because ICOP is a Sharia compliance manufacturing plant. That is another hurdles for us. When we want to produce our products, we have to make sure that all the ingredients that come to us with halal certifications or with a COA that confirm it is halal. That is another hurdles that we have to face actually as the Sharia compliance manufacturing plant. But as this is our duty, so we have no choice than to get the materials with all the requirements.
Dr. Siti, may I follow up with some questions?
Yes.
As you said, ICOP actually has no product for the herbal medicines apart from the eucalyptus oil ointment. What about the overall trend in Malaysia? So what is the most commercialized herbal medicines in Malaysia? Like what kind of plants, or is there a trend? Is it to certain type of disease or a certain type of pharmaceutical preparations?
Okay, just to share with you, nowadays, if you can see that we are very into like for drug abuse inside there, but recently we are into to approve the there is a for the drug abuse, I can't remember that plant for like ketum, all those things. I cannot remember what is the name of that. So all those that kind of like for mental health, things like that. For mental health, those are the for drug abuse, like to replace methadone, for example, like ketum. What is the name of that? I cannot remember the scientific name of that ketum. Mitri, I cannot remember that. But it's okay. We are. In Malaysia now they are talking about that, to the regulations to approve on planting that kind of plant, but then still on debatable lah. Okay, and then nowadays actually in Malaysia we are into using these phytochemicals for treatments, for cardiovascular, for diabetic. Actually, if Dr. Alfie, I think can give us a lot of figures on that. That diabetic is actually one of the second after second disease that is in Malaysia now. So most of the peoples, they don't like, most of the peoples in Malaysia, especially those in the village, they don't like to use the injections to control their diabetes. So they will go for herbs. The problem is the way they use the herbs is they cannot control. They stop using the modern medicines and they concentrate on using the herbs without guidelines from healthcare professionals. So meaning to say that actually peoples are going towards these herbal directions. It's just that that's why we at the regulatory or Ministry of Health, actually we are into the study of traditional herbs with therapeutic indications. We are trying to register the products, the herbal products with therapeutic indications, but then the method of doing it not as stringent as with pharmaceutical products. Like for pharmaceutical medicines, we need to have a BE study or bioequivalent study and all those things, which takes a lot of years to have the information whether the products can be used or not. But with this, I think for herbals with indications, they only need to use the in vitro. Maybe the one that Dr. Alfie is doing is enough to confirm that that particular products can be used, for example, for as anti-diabetic, or cardiovascular, or at the moment we can only label our herbal medicine to be used as with high antioxidant property. This general, cannot specific for certain disease. Okay, am I answering your questions?
Yes. Yeah.
All right.
But in the future, will there be say a more or updated guideline to specify because I think the whole direction is to make sure that herbal medicines can be accepted as the therapeutic medicines. So will there be a new guidelines or something like that?
Yes,
say,
it is in our guideline. Yeah, it is our guideline. It is, and it is now in the guideline already that I forgot to share with you all just now. In the guideline to say that we can there are certain methods or there are certain documents that is required when we want to claim that that particular herbals with certain indications. I think I can share later with your groups. Okay.
Oh yeah, very welcome if you do
right.
that.
Okay. Okay. Inshallah, I will do it. Okay.
Yeah. I think I would like to have some questions for Professor Alfie. Is that okay while we are waiting for you to find your the documents?
Yes.
Is that okay? Oh yeah.
Okay. All right.
Yeah. I'll get back to you very soon. This is a very simple question, Prof. Alfie. I think metabolomic approach is is a very good approach, especially an attempt to map out the biological effect and metabolites. of herbal medicine extract, given the abundance of metabolites in just one single extract. This is a quite tedious and lengthy process. So it is more like a high throughput screening, yeah? Is there an international collective library of the metabolomics? For instance, for the protein, we have the protein database or the genomic sequence database. So is there the equivalent for herbal medicines?
Okay, for the metabolomics, for the database, this is it's not like as good as protein. Yeah? So it's very we are lacking of database. This is why the Metabolic Society we try to help each other, yeah, to upload all the database. Yeah, if you isolate the compounds, they try to upload it. Another thing is 5% only metabolites has been characterized. Yeah, so the rest we never know what's that. Yeah? So it's always updated new metabolites, yeah, every year. So the thing is we try to do our best based on the reported one. Okay, so of course metabolomics is very interesting tools. Yeah. However, as you know that there is no one single instrument that can be used to analyze all metabolite. So we are like complement each other. One instrument is complement to another instrument. So normally I use at least, yeah, two instrument, GCMS and LSMS. So GCMS is for the very low molecular weight, and for the LSMS is a middle to metabolite, molecular weights of metabolite. Yeah. So if you said this is difficult, yeah, this is depending on our how we handle the instrument. Yeah. As long as we can have database, and as long as we can easily compare database, and then if we want to explain whole metabolites, which is not reported, so we cannot, only based on the reported one. Okay, I think can help you.
Thank you very much. Now, which is very curious, because if everyone tries to create the database from this, and that might be nice to have the international collective library, but and I think you said that there is all the researchers are working on it together. So yeah, that's a good start. We have another question for you. It's still from Budinat. Related to your slides, analytical tool based on the herbal fingerprint, how to apply its study in quality control of herbal products? Is it sufficient for quality control for herbs and not using the exact compounds which are related to the biological activity? Do you have any comments on that?
Yes, this is one of the tools, yeah, for the quality control. Of course, you can use another analysis, yeah. But I think this FTIR compared to other analysis, yeah, FTIR is quite simple and fast. And then I have already experienced this with different batch, yeah, and also the varieties of the plants is quite powerful. Yeah? So if you see that if even a single difference, yeah, of the metabolites, they can affect the the spectrum. So in FTR, we don't care what is the compound is, but as long as the fingerprint difference, then it will be different. So that's why we apply the statistic, yeah, in order to see the grouping of the pattern, yeah, of the spectrum. So by eyes, maybe you cannot see. By statistic, you can see. Okay. If we compare, for example, with the LSMS, the TLC. Yeah? TLC problem is the reproducibility. Reproducibility is quite difficult. If you want to see the chemical, the chemical region, is not reproducible. If you also use this UV light or others light, yeah, depending on the wavelength. Sometimes there are a lot of metabolites which is not in that range, so you can see as well. But if they are, you can catch up the metabolite. So this the nice from the FTIR. And another thing FTIR is because of this is the physical response. So this will be robust. If you compare to TLC, yeah, TLC always is simple, but they are depending on the chemical properties, depending on the solvent, yeah. So sometimes it's not also reproducible. It's depending on the company who made the TLC. So I really prefer using FTIR for the simple. I'm talking about simple and fast method. Maybe any more question?
Ibu Dina,
Yes,
to follow up on that.
Yes, thank you for the answer, Mr. Alfi. I also noticed that fingerprinting, yeah, one of the instrument you can use FTIR, and it is more handy to be applied as a quality control. And I guess, and I think it is a challenge for the Badan POM, it is an institution who which approve the sort of herbal standardization in Indonesia, and I think it is we can suggest them that this is an alternative method used instead of TLC, as you mentioned, with lack of the reproducibility. And yeah, we can propose this fingerprinting for an alternative method for quality control. I agree with you. Thank you.
Maybe for in additional information, FTIR only 1D. Now we have 2D FTIR. So it's also another thing. You can use the ATR. So you don't need the KBR. You just put this powder, very easy, and then you can 5 seconds you get all the results. So I published in vibrational spectroscopy, and they never reject me. Alhamdulillah. So it means that that is quite valid. Yeah, for in-house training is permissible.
Yes, this is a good information, I think, especially for application in Indonesia. Thank you, Mr. Alfi.
Okay, you're welcome.
Talking about robustness of the methods, is there minimum requirement, like maybe minimum 2 methods to show the robustness of the results of the methods as well as the type of the methods? Or is there any other parameter for the robustness?
Yeah, robustness, a lot of things. Yeah, it's depending on the reproducibility. If you, for example, one researcher, they analyze and then you have another result, then it's not robust. Yeah, because can you imagine this is TLC, for example, it's depending on, yeah, a long time ago, you know, TLC, we don't have this from the ready-made, right? We make ourselves. I remember when I was student, I used to make my own. So depending on a student, yeah, so they have different result. So now, we have the standard TLC product, but still, you know, if you have brand to another brand, may make a difference. And then another thing is, of course, we have the equipment now, yeah, to see the when the eluted, yeah, the solvent eluted. But again, yeah, when you stop it, when you stop the elution, another thing. So it's difficult. But if you have FTIR, just put there, have pressure. So the reproducibility is much better. So you compare analysis today, tomorrow, and it will be different. If you have TLC, for example, it will be like that. It's depending on solvent. Yeah, solvent, another brand, and then another things. So that's why if you want to establish method for the quality control, the quality control of your product, it's better to use a spectroscopy, not the chromatography, because chromatography depending on the stationary phase. So maybe you have SPLC. SPLC, one company say, okay, this is the best column. Another month, another one is best. Another's always best, right? So when you come back again another 2 years, that column not exist anymore, But we developed them 2 years ago, so you don't have anymore. No. This is now we have another best column, but best column is different result, right? But if you have NMR, if you have like FTR or UV, but UV is the different story. It's not like it's not as not specific, as FTR, So if you want to have this for the QC, better to use the instrument based on the spectroscopy.
Thank you very much for the insight. Dr. Siti, do you still want to share the guideline with us?
Just hold on, I go to the page. Session. Okay. Can you see anything?
Yeah, we just gonna.
Right. Actually, this the documents that I have still in the confidential, but it's okay. We can always share, no problems. Okay, because it is already in use already. So here is extension of alphabets for natural product with therapeutic claim. So the session number of usually we have the ML number, and then followed by 8 numbers ending with the letter B. So okay, if you look here now, when we are preparing the products, we can already have the therapeutic claim for our products. For examples, like the product is used for whatever claim that we want. But how to do it? Actually, it is not very clear yet. For example, like Herbal here, efficacy, a relative concept referring to the ability of medicines or treatment to achieve beneficial clinical effect. This may be measured or evaluated using objective and subjective parameters. So herbal medicines include herbs, herbal materials, herbal preparation and finish, herbal products. For example, they must have the herbal ingredients, herbal materials, and what are the herbal preparations that is required, and then the finished herbal products. And then the scientific evidence, when we want to claim, you must have a quantifiable data, and usually includes reports of clinical trial in humans or human epidemiology study, animal studies, and other cellular or pharmacological study due to the quantification nature of scientific evidence. Scientific indication and imply clinical efficacy where the indication is supported by such data. So it is not here says that you require the bioequivalent study. So that is what I want to share with you all actually about this evidence that is required. I mean, in order for you to claim the products that you need to have, you can do the herbal products with therapeutic claim. Of course, you still need to have the clinical, you need to have data, but then it is not necessary bioequivalent study, which is very expensive.
I agree with you. Yeah.
All right.
Thank you. Is there any other questions from the attendees? All the students, you guys are welcome to give questions for us. Dr. Siti, what is the most challenging part in the standardization of raw materials?
The most challenges part, because we never do the raw material standardizations, but I think in finding the raw material standardization is the in order to get the full informations about the documents on the raw materials. For example, the constitutions, because sometimes most of the supplier, they do not provide us with the full informations, as I told you just now in our slide. What are the requirements by the WHO when we want to, because I think as discussion just now, it's not easy to get each of the constituent, the percentage of each of the constituents in the plan. But inshallah in future, Dr. Alfie can do it.
Yeah, we're waiting for its results to come out, and then we can use it for the standardization.
Yeah. We have to work with these people. So.
Yeah, I think this is an integrated process from the analysis, and then we can apply it in industry. Yeah, it looks like there are no other questions, and we have covered all other questions as well that we have so far. So is there anything else that Prof. Alfie or Dr. Siti want to add before we close the discussion? Like maybe some concluding remarks. Oh, sorry, there is a... Idina, you raise your hand? Yeah.
Yes, while waiting for others question, maybe I can ask more question for Prof. Alfie. Prof. Alfie, it is metabolomic study is now also many researchers try to apply metabolomics nowadays, especially in Indonesia as well. And I wonder how to deal with the metabolomic study. Is it similar to what we have in bioassay guided fractionation, or we need include certain circumstances to do the metabolomic study?
Yes, thank you, Budinar. Okay, I talk regarding the bioassay guided fractionations. Yeah. So in bioassay guided fractionation, meaning that when we do the fractionation, we just follow when we select the fraction, we should based on the active fraction. So if you have, for example, from the column, you have 12 fraction, and you want to select among the 12 which one we should go further. So we need to test the activity, each of the fractions. So the problem is sometimes we are missing the bioactivity. It happened to me when I did my master. So I waste my 6 months because losing the bioactivity. Actually, when you have this raw material, this is very, very active and promising. And then after I did the open column, all missing, no activity at all. So then we are just want to figure out, so where is the compound? Where is the... Actually, if you mix them again, you remix them, active again. You see, there is synergism effect. So in this case, then you will never found the bioactive compound. So you are already missing the activity. So just stop. Yeah, then what I did is I changed my herbs after 6 months. But if I apply metabolomics, it will not happen. Okay. Once I know the activity from the extract, which is very, very strong, then just continue. So I from beginning, yeah, I will know, okay, the peak with related to activity is, for example, peak 1 until 10, yeah, for example, right? And then when I do the metabolomics, now we call as metabolomics fractionations, yeah. Okay, meaning that when you fractionate, you did not follow the activity. you follow the peak that already identified and not already selected using during the metabolomics mapping. Yeah. So, for example, through the metabolomics mapping, I found the peak, which caused the activity is peak 1 until 10. Others peak not important. So when I do fractionation, I just follow where is peak 1, where is peak 2, where is until 10. Okay. If you find out, okay, peak 1 until peak 5 is reported already and already commercialized in market, so why you need to isolate? Yeah. So, but peak 6 to 10, for example, no reports reported, and or there is reported but not available in the market. Okay. So, but you want to test to your activity, then what you will do is metabolomics fractionation is you just analyze fraction 6 to fractions the other fraction. You just test if there is peak 6 to 10. Okay, and then you go forward until you get the pure compounds, which are should be the peak 6 until 10. So we do at the time we don't know what what is the structure. So that's why you go to NMR, you go to MS in order to elucidate the structure, and it should be maybe 90% of this compounds. The possibility will be the novel compound. So no people report this compound before. So that's why, in terms of publication, it will be better, right? Because we will not waste our time after 2 years and get, okay, I will get quercetin, so already there, or I get the I remember my work, my master, para-hydroxybenzoic acid. So what for? Already there. So what we want is really novel compounds, right? So that's why now we propose the name is not is metabolomics guided fractionation now. So my student already work on that, and we will publish. Hopefully it will be the first that we write as metabolomics guided fractionation. Who knows? Yeah, I think it's quite a potential. It's very nice. Yeah, it works. Yeah, inshallah.
Yes, it's a very good idea to have a metabolomic guided fractionation, Mr. Alfie. Yes. Yeah. And I wonder how to deal with the sensitivity of the tools. If you use NMR, do you use other what is it? Instrument to deal with? Usually the minor compounds cannot be detected by NMR.
Yeah, NMR actually is for okay, the NMR is of course is another advantage, but it's not sensitive. So imagine that you have minute amount you cannot detect, but LSMS possible, it's quite sensitive. That's why if you have possibility, if you have, for example, limited budget, research nowadays is quite limited to COVID, right? So the first priority, according to me, first priority is LSMS. Secondly is GCMS. Last is NMR. But if I am rich, then I will analyze all, LSMS, GCMS, NMR, all, because the problem with LSMS depending is ionizations. Some of compound not ionized, as well as GCMS. Although you have the refractize it, it cannot be ionized. So this can be measured by NMR. Yeah, of course, maybe NMR not sensitive, but who knows? Yeah, this can the one which is not ionized is something can be seen by NMR. So it just help. It can help.
Yes, Mr. Alfie. In other words, it is better to use two instrument both probably LCMS and NMR
Yes,
to complete each other. Yeah.
Yeah. Sometimes if you they can confirm each other. For example,
Yeah.
you have LCMS, you can see NMR. Yeah,
Yeah,
there are
yeah.
NMR should be there. But for example, LCMS, because they are very minor, they can detect a minor amount. But in NMR, sometimes what you can see is they overlap with others. So you cannot see the peak because of overlapping. Yeah, although you already have the coupling result, you still cannot see because they are very minute amount. But if you have the major amount in the sample, yes, you can see. Yeah? But maybe sugar, but that one is not active. You can see, right? You can see it will not MDA will not pinpoint this is the active compound. Yeah, right.
Yes. That's the art working with the natural products. Yeah, we deal with hundreds of metabolites. Thank you for the discussion, Mr. Alvin.
And in addition, yeah, NMR is very good for to use for the quality control. Say maybe Rob always said this one, right, Rob? Okay, he compare NMR with LCMS. NMR, if you have NMR, it will stable another 100 years. But if you have LCMS, no, yeah, because this is using the column. Okay, but in order for us to know the compounds, to identify the compounds in new compounds, or then you must use LCMS because NMR difficult because not sensitive. But if you want to use LCMS as your quality control instrument, I think this is quite it will not recommended, yeah, because the result can be different from year to year. Okay, that's why if you quality control, just use the spectroscopy. Yeah.
Thank you, Mr. Alvi.
Okay, welcome.
Thank you, Budina,
Perhaps
for the discussion.
Wirna, I have a question for Alvi.
Yes, please. Yes, please.
Okay, I
Kakak
think
Safono.
just a simple question for Alvi. As interesting, I'm interested to your statement about that.
Yeah.
The advantage of spectroscopy and then chromatography. That spectroscopy approach have advantage, and then chromatography based on as you explained before. But as we know that for the application of instrumental analysis, it is mostly we use a hypnated instrument. It means that we combine between chromatographic approach and spectroscopy approach. But I agree that for qualification for quality control, and you have clarified why the spectroscopy approach is better, because the data will be more stable for long than the data from chromatography, yeah? Because there are development in the column and etc. So I just want to I want to confirm that the advantage of spectroscopy approach is just for the not also for the practical advantage than the accuracy, for example.
Yeah, yes. For spectroscopy, we can use the data for the long terms. Yeah. So because once it's also not easy, yeah, for us to establish the model, and then suddenly, yeah, when you establish, and then the supplier said, "Oh, no column anymore." So we we developed a new calibration model. This also takes very long time, because when you have the calibration model in terms of the bioactivity, meaning that you need to test each of the sample to test to the bioactivity again. This is also costly. So that's why for the quality control, the simplest one is the FTIR, as I suggest. But if you have NMR, it's also nice, because NMR is also fast. Actually, if you dissolve them in the MOD, for example, or water or mix of that, then you can get it easily. 5 minutes, maybe you scan 1 to 8, and you get the spectrum. But then it's also expensive, right? Because you need the MOD, you need NMR is expensive, So for the company, small company, I think FTIR is quite okay. Last time I already tried to compare between this FTIR and NMR. It's not bad. FTIR, of course, NMR is better, but not significantly, significantly different. But it's still acceptable FTIR. Because NMR, you can see they are not as overlap as FTIR. FTIR, we have overlapping, broadening peak. NMR is much better. But when you compare between the calibration model, it's quite okay. The R-square that I get is more than 0.9. It's fine. That's all. I think we are talking about the cost, right? During the analysis. And if analysis is too high, too expensive, then the price of products will be expensive, right? Thank you, Dr. Sabtono.
Okay, thank you, Prof. Raffi.
Thank you, Pak Sabtono. I think we have come to the end of our discussion, and I would like to invite everyone for a photo session. Please follow the IT team to get ready, and for everyone to turn on your video camera so we can have all of you in one screen. Please, for the IT team, let us know when you would like to take picture of us so we can get ready.
Baik, untuk semua peserta bisa menyalakan kameranya terlebih dahulu. Baik, siap-siap. Saya hitung 1, 2, 3. Sudah, Bu Erman.
Thank you. Thank you. Okay, thank you, everyone. We appreciate you being here in our webinar, especially to Professor Afi Khatib and Assistant Professor Dr. Siti Hadijah Syamsuddin. We have learned a lot about herbal medicines from the metabolomics approach and then the development and commercialization in just a very short time from your lecture. So this is a priceless experience to listen to your talk. And for all the attendees, thank you for the active participation. And just a quick reminder to fill out the attendance form, and that there will be next webinar series. So please ensure to keep updated for the upcoming series. Thank you again, and see you in the next series. Thank you, Prof. Alfie. Thank you, Dr. Siti. See you.
Okay, thank you very much. See you all again.
Thank you,
Bye-bye.
Prof. Raffi. Thank you, Prof. Siti.
Prof. Alfie.
Okay.
Terima kasih ya.
Thank you for the invitation.
Prof.
Okay.